Hi and welcome back to another video. In this video I'll take you guys through nutrition in plants, specifically autotrophic nutrition, which basically means self-feeding, and the organisms involved in this type of nutrition make their own food, as most of us know. And under autotrophic nutrition specifically, I'll talk about photosynthesis, and I'll make sure to reveal everything you need to know about it at A level. So from our equation, photosynthesis is the process by which plants make their own food using carbon dioxide and water in the presence of light trapped by chlorophyll. Now, plants and other photosynthetic organisms such as green algae and photosynthetic bacteria are able to capture energy from sunlight and use this energy to synthesize complex molecules such as glucose from carbon dioxide and water. Now, at all level, I don't think there's anything complex you were taught on photosynthesis apart from the definition, factors affecting photosynthesis, and maybe the word chlorophyll. However, at A level, you actually go into more detail on photosynthesis. Okay, now although leaves are the main sites of photosynthesis in most plants, it can actually take place in any part that is green. But I'll use a leaf. So our leaves obviously have plant cells, and these plant cells have organelles called chloroplasts. And these are the sites of photosynthesis. So photosynthesis takes place inside these organelles called chloroplast, and I'll briefly talk about its structure. Okay, now there's our chloroplast, and the chloroplast has a double membrane which controls what enters and leaves the chloroplast. Now inside the chloroplast is a fluid-filled matrix called the stroma, and the stroma has a number of structures inside, which are such as cellular DNA, 70S ribosomes, starch granules, and so many others. But what you need to know about the stroma here is that it's the site of the light-independent stage of photosynthesis. I shall talk about it as we go on. The next part is the grana, and these look like a stack of coins, and each of those stacks or disc-like structures are called thylakoids, and these are sites of the light-dependent stage of photosynthesis. Now, the thylakoids have a thylakoid membrane in which I embedded what we call photosystems. There's photosystem 1 and photosystem 2, and also embedded in the thylakoid membrane is the electron transport chain, which consists of a series of electron carriers, and inside the thylakoid space are enzymes and other molecules needed to make ATP and reduce NADP, such as ATPase. Now, I talked about the thylakoid having photosystems, and these are two. I've already talked about that. Now, these photosystems are actually very similar, and they contain photosynthetic pigments such as carotenoids and chlorophyll. There's chlorophyll A and chlorophyll B. Now, the photosynthetic pigments here serve one role, and that's to absorb light energy, and they then convert it to chemical energy, and they allow absorption of different wavelengths of light. So this is our light spectrum, and as sunlight falls on a plant, some wavelength of light are absorbed while others are reflected. Now, the pattern of absorption and reflection depends on the pigment, and our photosynthetic pigments, as already told, include carotenoids, chlorophyll B, and chlorophyll A. Now, carotenoids are yellow, orange, and red, and are known as accessory pigments because most of the light they absorb is passed to chlorophyll B and later chlorophyll A. So, carotenoids strongly absorb in the blue and the blue-violet range, or between 450 to 500 nanometers. And chlorophyll, which is more abundant, absorbs strongly in the blue-violet range and orange and red range, or between 450 to 500 nanometers and 650 to 700 nanometers. Now, this graph shows the action spectrum and the absorption spectrum, and the action spectrum shows the effectiveness of the different wavelengths in light in stimulating photosynthesis. So the action spectrum here, the peaks show the wavelength of light that effectively stimulate the process of photosynthesis, and the other graph is the absorption spectrum, which shows the relative amounts of light absorbed at different wavelengths by a photosynthetic pigment, let's say chlorophyll. So you realize that more of the blue-violet light and the orange and red light are absorbed by our photosynthetic pigments during photosynthesis. Now, I said the pattern of absorption and reflection depends on the pigment, so you realize that most of the plants are green because the green color in the light spectrum is not absorbed, instead it's reflected. That's why you see the green range of light is at its lowest in both our graphs. So I don't take much time on this. If you feel like I missed anything on those two graphs, you can go and do more research, because I know there might be more. to discover here. Now, I talked about photosystem 1 and photosystem 2, which are embedded in the thylakoid membrane. So I already told you these are very similar to each other, and they have photosynthetic pigments, carotenoids, chlorophyll B and chlorophyll A. Now I want to hit on something here, and as light energy is absorbed or trapped by a photosynthetic pigment, it passes from the carotenoids to chlorophyll B and later to chlorophyll A. So in our photosystem is something called the reaction center. Now the P there stands for pigment, and the 700 and 600 stands for the absorption peak, or the wavelength that is mostly absorbed by our reaction center, since it's a modified form of chlorophyll A. Now P700 and P680 are found in photosystem 2 and photosystem 1 respectively. So as light energy is transferred all the way to the reaction center is photoactivated or excited, and this causes electrons to also become excited. Now explaining this would be very confusing, so I'll explain it under the light dependent stage of photosynthesis in what is known as the Z scheme. So I'll summarize the light dependent stage in what is known as the Z scheme. So here, I hope you can see everything very clearly, the photosystems and the reaction centers. So when energy from light is transferred to the reaction center is photoactivated, and I'll start with the reaction center of photosystem 2, which is P680. Now when the reaction center is photoactivated, electrons are boosted to a higher energy level, and these electrons are then received by plastoquinone, which is an electron acceptor. So these electrons then pass from our electron acceptor along a series of electron carriers, or they pass along the electron transport chain to photosystem 1. So as the electrons move from one electron carrier to another, they keep on losing energy, and the energy that is lost here is used to combine ADP and an inorganic phosphate to form ATP, a process called phosphorylation. But since it requires light, it is called photophosphorylation. Now I'll talk about photophosphorylation and how ATP is made in a minute. I'll explain what really happens there. But for now, we shall go back to photosystem 1. So when light is trapped by chlorophyll and transferred to the reaction center of photosystem 1, its electrons are also boosted to an even higher energy level. So the electrons are then received by our second electron acceptor, which is called ferredoxin. Now when an electron is lost, the photosystem here is oxidized, and when an electron is gained, the photosystem is reduced. So you should remember that. So when photosystem 2 loses an electron, it causes some sort of deficiency. Since it has lost an electron, its electrons are then replaced by water, and this is by photolysis of water, which takes place in the presence of light. So here water splits into protons or hydrogen ions, electrons, and oxygen. The oxygen is released as a waste product, so it's released during photosynthesis. Now the electrons from photolysis of water replace those that are lost in photosystem 2, and photosystem 2 is then reduced once again. So you should remember this is some kind of oxidation and reduction taking place, some kind of redox reaction, and it's just continuous. So the electrons lost from photosystem 1 are replaced by the electrons that move from plastoquinone along the electron transport chain to photosystem 1. So back to ferredoxin. Now as the electrons move from ferredoxin, these move along another series of electron carriers, and these electrons combined with the protons from photolysis of water, and the electrons and protons or hydrogen ions are taken up by NAD, which is a hydrogen acceptor, forming reduced NAD, and this reduced NAD is used in the light independent stage of photosynthesis. Now I promised to talk about photophosphorylation, and this is the process by which light energy is used to produce ATP from ADP and an inorganic phosphate. And how this happens is that as electrons move along the electron transport chain from plastoquinone, please, I intend to give you guys the picture here. So As electrons move from plastoquinone along a series of electron carriers to photosystem 1, they lose energy. So if this is our thylakoid, remember all this is happening inside the thylakoid. So our thylakoid has a thylakoid space, a thylakoid membrane, and our photosystems. So some of the enzymes in the thylakoid are called ATPase, and this is the most important one here. So as electrons move along the electron transport chain, they keep losing energy, and this energy is used to transfer hydrogen ions or protons across the thylakoid membrane and into the thylakoid space. So as this happens, you realize that protons keep on building up inside the thylakoid space, and the inner membrane of the thylakoid is impermeable to these protons. So they can only flow out through a specific point, which is permeable to the protons, and this is called the ATPase synthase complex, which contains our enzyme ATPase. So as these electrons move through the ATP synthase complex, which contains ATPase, remember, they provide energy to combine ADP and the inorganic phosphate to form ATP, a process called photophosphorylation. Now I've tried to explain that, but I know it's a bit confusing, so you might have to do a little research on that. Now back to photophosphorylation. It is divided into cyclic photophosphorylation and non-cyclic photophosphorylation. And I will start with cyclic. I'll start with cyclic photophosphorylation. Now this only involves photosystem 1. When light raises the electrons in the reaction center of photosystem 1, it is photoactivated and its electrons are boosted to a higher energy level, and these are taken up by the electron acceptor, in this case ferredoxin, since it only involves photosystem 1. Now these electrons are then recycled back to photosystem 1 via another chain of electron carriers, and what happens here is that it generates sufficient energy to combine the inorganic phosphate and ATP to form ATP, and no reduced NAD is made during the cyclic photophosphorylation. Then there is non-cyclic photophosphorylation. Now non-cyclic photophosphorylation involves photosystem 1 and photosystem 2. So the electrons are excited in photosystem 2, taken up by electron acceptor, move through the electron transport chain, producing ATP, and these electrons then they then move to photosystem 1. Now the electrons in photosystem 1 are also activated, and these are then taken up by ferredoxin. Now these electrons pass down a chain of electron carriers and combined with protons from photolysis of water, and these combined with NAD, which is a hydrogen acceptor, to form reduced NAD. I already talked about that. Now from what I've explained, you should be able to compare cyclic and non-cyclic photophosphorylation and photosystem 1 and photosystem 2. You can do some research to get more insight on that. So you realize that during the light dependent stage, ATP and reduced NAD are formed to be used in the light independent stage. So that's all you need to know on the light dependent stage of photosynthesis. Now with that said, we can turn our attention to the light independent stage. Now it's called the light independent stage not because it takes place at night, but it does what this means that it does not actually require light to take place. So it does not necessarily take place at night, but can still take place during day, only that it does not depend on light. And then I already talked about where it takes place. It takes place in the stroma. Now there are two pathways of the light independent stage. One is the Calvin cycle, which is common to C3 plants, and the other is the Hertzsprung pathway, which is common to C4 plants. And I will start with the Calvin cycle, which takes place in C3 plants. Now they are called C3 plants because the first product of photosynthesis here is a 3-carbon acid called glycerate phosphate. The Calvin cycle involves 3 main stages, that is carbon fixation, the reduction phase, and the regeneration of the carbon acceptor. And I'll start with carbon fixation. So the carbon dioxide from the atmosphere diffuses into the leaf and into the plant cells, eventually into the stroma of the chloroplast. So what happens to this carbon dioxide is that it combines with the 5-carbon acceptor called RuP, or in full it's called ribulose bisphosphate, and that forms a 6-carbon compound which is unstable and immediately breaks to form 2 molecules of GP, or glycerate phosphate. Now this all takes place in the presence of an enzyme called RuP carboxylase, or Rubisco. Then next is the reduction phase. Now during the reduction phase, this GP, which is a 3-carbon acid, so it contains a carboxyl group, and this reduction requires hydrogen from reduced NAD and energy from ATP. So in the end, since it's a carboxyl group, those who are doing chemistry will get this more. It loses one oxygen atom, and that becomes an aldehyde group called and this is called triose phosphate. So triose phosphate is the first carbohydrate formed during photosynthesis. And lastly, on the Calvin cycle is the regeneration phase of the carbon acceptor. So what happens here is that most of the TP that is formed is used to regenerate group P, which is consumed in the first reaction. So what happens that during the first reaction is that when group P combines with carbon dioxide form GP, it loses one phosphate. So it's no longer called ribulose bisphosphate. Since it has lost one phosphate, it's just called ribulose phosphate because it has lost one phosphate. So what happens is that most of these TP is used to reform group P, and this requires energy from ATP. So when the ATP breaks down, the inorganic phosphate from ATP combines with this ribulose phosphate to form ribulose bisphosphate again, which has two phosphate molecules. I hope you are getting that. And that enables the cycle to continue. So the remaining TP is then converted to other products such as carbohydrates. So this is where the remainder of the ATP is used. And that is that on the Calvin cycle. Now I'll talk about the Hatch-Slack pathway, which is common to C4 plants. Now they are called C4 plants because the first product of photosynthesis here is a 4-carbon acid called oxaloacetate, and these C4 plants include most tropic and subtropical plants such as sugarcane and maize, or even corn in some areas, and so many others. Now C4 plants possess a unique leaf anatomy in which 2 rings of cells are formed around each of the vascular bundles. So the two cells here are called bundle-cyst cells and mesophyll cells, and these have chloroplasts which differ from each other. Now here, carbon dioxide and hydrogen are transported from the mesophyll cells to the bundle-cyst cells. Now once in the bundle-cyst cells, the carbon dioxide is released again, and the C3 pathway occurs, or the Calvin pathway takes over. So that Slack pathway also has 3 main stages, which is carbon fixation, then we have the Mallet shunt, and the regeneration of the carbon acceptor. Now the Hatch-Slack pathway has 2 carbon acceptors, that is phosphophenyl pyruvate or PEP in the mesophyll cells, and RUP, which I already talked about, in the bundle-cyst cells. Now carbon dioxide is fixed in the cytoplasm of the cells by PEP in the presence of PEP carboxylase, forming oxaloacetate, which is a 4-carbon acid. Now the oxaloacetate is then converted to malate. To be specific, the oxaloacetate is reduced to malate using the hydrogen from reduced NAD, and that's how malate is formed. So the reduced NAD is then oxidized to NADP. Next is the malate shunt, and here you realize that the malate that is formed is then transferred through the plasmodase matter to the chlorophyll of the bundle-cyst cells, and in the bundle-cyst cells the malate is then converted to pyruvate, which is a 3-carbon acid. And the reason why it is now a 3-carbon acid is that during its conversion, the malate loses a hydrogen. So the NAD here, the NAD takes up a hydrogen from the malate. So reduced NAD is formed again, and it also loses carbon dioxide, and that's how it forms a 3-carbon compound called pyruvate. So from here you realize that in the mesophyll cells, carbon dioxide and hydrogen are added, and in the bundle-cyst cells, carbon dioxide and hydrogen are removed again. So this basically moves carbon dioxide and hydrogen from the mesophyll cells to the bundle-cyst cells, and that brings us to the last phase, which is the regeneration phase. But before that, the carbon dioxide is then taken up by RUP, and this enters this Calvin cycle, forming glycerate phosphate and later triose phosphate. So that's the fate of the carbon dioxide here. Now the pyruvate is transferred back to the mesophyll cells, and it is used to regenerate PEP using ATP. So ATP is broken down into ADP, an anorganic phosphate, releasing energy. However, the phosphate is added to pyruvate to form PEP, or phosphoenolpyruvate. So the inorganic phosphate is added to pyruvate to form our carbon acceptor, which is PEP. And this allows the cycle to continue. Now, I hope you guys have got the point there. And that is all I had on photosynthesis, and I believe that this has been of great help to most of you. And as usual, it's very important that you also do your own research, because that actually plays a bigger role than these videos. And when you get to know more on this topic, the next time you walk around outside and look at plants around you, not all of them, but most of them, you will try to appreciate the crucial role they play in the web of life that includes even us as humans. And I cannot emphasize this more. Please like the video, just like the video, and subscribe to my channel. And remember to hit that bell icon to get notifications every time a new video is released, because I'm soon releasing a video on C3 plants and C4 plants, limiting factors and compensation points. With all that said, thank you for watching, and see you in my next video.